Hello, well, good afternoon. My name is Sebastián Urquiza, and today I will talk about the work that we're developing in our lab, that is co-expression analysis coupled with gene regulatory networks, reveal master regulators in mouse heart induced by angiotensin. I work in Chile. I'm doing my PhD in biochemistry, and in our lab, we're really interested in knowing which are the key molecules that govern the gene regulatory networks. The gene regulatory expression, and also which are the new or novel potential therapeutic target. But before I introduce or make the main findings of our work, I want to contextualize which are the main purpose of this is to understand the role of angiotensin 2 in the cardiovascular disease. Angiotensin 2 is a vasoconstrictive peptide that is belong with the renin-angiotensin-aldosterone system that principally regulate the blood pressure in different in different cell types, specifically in the endothelium, that regulate or specifically constrict the arterioles or and to to augment the different blood pressure. But this peptide have a lot of outcomes in the body. So in different kinds of, pathological context, we have like rot generation and endothelin, and also the the secretion of other hormones in the body. So when you have different risk factors like cardiovascular disease, this neurohumoral dysregulation, we also find this angiotensin to highest level. But mainly in the heart, we found these two main type of cells, that is fibroblasts and cardiomyocytes. That is one that make the fibrosis process, and the ones that is cardiomyocyte, that is related to the pumping of the heart. And when you have this angiotensin 2, when you stimulate with the different type of receptors, you will look different outcomes that are related to, in a pathological context, you will have a excess of extracellular matrix production, and in the cardiomyocyte, you will look at the hypertrophy context in the pathological way. So this developed all these group of phenotypes. It called that cardiac remodeling in the pathological context that I was mentioning in the before. But if you look in the fibrosis or the in the cardiac hypertrophy, which one start at the beginning and start first, we are like looking more closely, specifically in the cell type. So if you look or you remember in the cell signaling class, with I really don't remember this pathway, but I also remember that we have two types of receptors on these cells. That is mainly the AT1, AT2, and it's governing the hypertension context. Is the AT1 responsible to all this cascade of different factors, and also calcium and other phosphatase. And in our lab, we are interested to understand which are the main factors that were related to this process, specifically transcription factors, and why we're really interested to look at this candidate, because and we are really interested in study cardiovascular disease, because I'm from Chile, and Chile has this problem of cardiovascular disease. In Chile, is the first cause of death. A main risk factor of high blood pressure is specifically 36% of the population have hypertension, and it's a silent pathology, because the only way you have to discover if you have hypertension or not is you measure your blood pressure. So, and we are looking for new pharmacological targets against early stage of cardiovascular disease, specifically pathological cardiac hypertrophy or fibrosis or something like that. So our main goal was identify master regulators, that is the key molecules that orchestrate changes in gene expression in response to angiotensin 2 in heart. So how we can afford this problem is in our lab, we develop different bioinformatic tools, but when we are like looking for a specific tool, we decided to combine and have two different approaches. It's like a system biology approach using co-expression analysis with coupled with gene regulatory networks. If you remember, co-expression analysis look for specific genes that modulate or have the same expression patterns. And when you look in another strategy, the gene regulatory networks, as Dr. Alberto was saying before, is like the one that you like looking at specific maps of interaction. So when you have these two approaches, but when you combine them, you could look at the specific gene co-expression models and look at the specific gene regulatory networks of those models. And why it is so important to look to these models and these interaction models? Because you will find some have genes that were like huge connections of these genes. Also, differential co-expression analysis based on you have different thresholds of the expression values. And we are interested in guilt by association prediction. So we took these genes or the specific model co-expression models and look which candidate are like tend to be more related to our phenotype, specifically in the heart context. So how we decide to approach this in cardiovascular disease? We use this bioinformatic workflow coupled with co-expression analysis with gene regulatory network. Is this transcriptomic data? We will look in the SRA, and we took this specific RNA-seq study that have these mice that have a stimulation with the angiotensin 2 peptide with during 7 days. You will know we have this pre-processing data with quality control and also with the genome alignment, and then we look on the read count with different programs like feature count, and then different expression analysis of you supposed to look up in the specific tools like volcanoplot or some visualized tools. And this is our approach to co-expression analysis and gene regulatory network construction. We use a semi tool that is a tool developed by our lab that specifically makes some co-expression models, and then we look at the specific gene context, gene regulatory network, and control and the angiotensin 2 model. And finally, we look at which are the main go terms or gene ontology terms that were involved in these master regulators. So this is the network is like not too quality, but the ones that really look up usually these networks, we look at specifically the transcription factors only in the context of angiotensin 2, and also we looked at 19 transcription factors that were involved here, and how many of them are reported in the literature, and which are the main master regulators of that. So if you look here, it's the known transcription factor affected in angiotensin 2, and also the transcription factors that will appear in the context-specific network, and the master regulators. There's like I will show you the list, and there's the 10 master regulators that were involved in this angiotensin 2 context, and there's one like is emerging, the ones that were reported, and the one we have found here, that is STAT3. Well, this is not well, it's a well-documented master regulator here that is implicated in the cardiofibrosis and hypertrophy as well. And we look and design this gene regulatory network, specific the models that we found. They were found 4 different co-expression models. And this model are interesting because in the model 1, we have this well-done documented that the fibrosis and extracellular matrix remodeling are the main gene regulatory process that we found here, and also collagen containing extracellular matrix. Well, it's like not new, but we're like specific models that these genes are involved in this M1. And the ML2 is surprisingly, we look at several genes that were cell cycle and proliferation. So we looked at cell cycle regulation, chromosome segregation, and mitotic process were involved in this model. So we're like interested in why these processes are so enriched in this model. So we look the master regulator network, and here's the 10 master regulators that were involved in this. And the biological insights that were discovered here is promoting inflammation and fibrosis, RELA, STAT3, and Jun and FOS contributing the expression of this phenotype in angiotensin 2. So it's a very well-documented relationship, and also mediated hypertrophic growth. Well, I will move forward with that because the main finding is this network is like MYC were down-regulated, and previous report say that MYC was upregulated in angiotensin 2. So we're looking in the specific model of angiotensin 2. It's like a premature exposure of angiotensin 2 will lead to down-regulated on MYC because it's like involving in the specific proliferation and cell programming function. So balancing also cell survival and apoptosis, we also look up the TP for 53 and STAT2 is like co-expressed and co-regulating in this different type of process. So for the final remarks or takeaway message, using co-expression analysis coupled with gene regulatory provides a robust framework to look at specific transcription factors in a specific condition. Also, this work reveals critical molecular process like fibrosis, inflammation, and cycle replication that were not well documented, but we will describe in further investigation or research. And this finding provide a roadmap potential therapeutic target because there's a 10 novel master regulators in here. And the knowledge, this is we are a really big lab in Chile. All the work is for them, and all the contribution that we make and practice on all this presentation. And lab members, also the funding, also, well, not all this money, all this support is emotional support for these people, and all the co-authors that will help me for doing this work. Thank you very much.